All right, today we're talking about calcium. My name is Jack. I'm the founder of Cultra, a nutrient analytics platform for crop consultants. Today with calcium, we're going to go over three things. One, what calcium does in the plant. Two, how it's uptaken by the plant. And three, how it interacts in the soil and how we can manage it. So first, what calcium does in the plant. It has two primary functions. So calcium's first role in the plant is cell wall integrity. It provides the structure of all of the cells in a plant to be resistant against outside attacks and just gives it overall strength. Calcium interacts with this thing called pectin, so a polysaccharide. Basically, it helps make this structural component kind of like concrete. So it interacts and makes these cross links with this pectin to give your cells in the plant, especially in things like fruit, there's a lot of structural integrity. This is really important when it comes to disease and pest pressure because what diseases do is they try and infiltrate this cell wall. So a pathogen will inject an enzyme where it'll try and that will break down. You see this in like blossom end rot and stuff like that with tomatoes. And having robust calcium cell walls help prevent the pathogens from being taken into the plant. So think of it like a line of defense. Now, this cell wall integrity has a couple other pieces. Another piece I want to touch on is its interaction with boron. So when calcium's interacting with pectin, this polysaccharide, it's kind of like pouring concrete. But there's a second very important piece that has to do with boron. And so boron interacts with another chemical that's in the plant, something similar to pectin but has a really long fancy name. And basically what that does is it makes the rebar that goes into the concrete, like we see in construction. So the calcium does the concrete, and then the boron is specialized in making that rebar structure. So we get both flexibility and firmness in our cell wall, which is very important when we are talking about things like fruit quality, fruit firmness, and fruit shelf life. So in addition to the fruit cell wall integrity, it also is important for all cells in the plant. So all of the cells, especially the external cells in the leaves and even in the roots, can be help with preventing pathogen attacks. So that's like that first line of defense, that wall. A cell wall that has abundant calcium is very important for making sure that we have a strong first line of defense against a lot of these pathogens that try and infiltrate the cell and kill it. And so calcium helps prevent that. The second very cool piece that calcium does is it's responsible for this thing called the internal second messenger. So calcium sits inside what's called the cytosol of the cell. So if you have a cell and you have a bunch of organelles, there's this gel substance kind of all around it. And there's free calcium. It's written like this: calcium in the cytosol. I think that this is the coolest thing ever. So the cytosol calcium, the calcium responsible for being this internal second messenger, has its own language. It's responsible for communicating to the plant the things that are going on in the plant and communicating up and down what's happening. Things like mycorrhizal fungi being attached to the roots, or a defense coming in, or if there's excess sodium at the roots. This calcium sends messages to the plant as this internal second messenger. And the way it does this is through its own Morse code-like language. You might be thinking, like, that doesn't make any sense. Like, that's insane. And that's what I thought when I read this research paper. But what it has is you have a small amount of this cytosol calcium in every cell. And when something happens, it'll increase the amount of calcium and then let it go at different rates, kind of like Morse code, like on, off. You have your calcium level in the cytosol, and then it says time. And if the calcium looks like this, it says one thing, like if the plant is being attacked, and if the plant is trying to get some mycorrhizal fungi, like it notices some mycorrhizal fungi attached to the roots, it could look like this. And so you can see that this frequency of calcium influx and outflux in the cell is responsible for communicating with the plant. And so I just think that's the coolest thing ever. So we got two primary things that calcium does in the plant. The first one, cell wall integrity. The second one being this internal second messenger, acting like this Morse code within the cell of the plant. So both of those are incredibly important. Now, when we're talking about cell wall integrity, when we're entering into fruit production, right? So there's a short window of time where the fruit sizes up. This calcium, we want it to make consistent, strong bonds. Now, the bonds that calcium makes, as opposed to the bonds that that boron makes, are slightly different. So calcium bonds in a cell wall are electrostatically connected, right? So what that means is there's a negative and a positive charge, and that's what holds that wall together. But when it's working with boron for cell wall integrity, it has a covalent bond. So that's makes the rebar, and the electrostatic connection with calcium is what makes that concrete. So that balance between flexibility and firmness. And when our fruit is growing, we want to make sure that the cell walls, when they're being built, that the plant has an abundant amount of calcium able to be taken up as that fruit is enlarging, and each of those cell walls gets built properly. I like to think of it as you want to have bricks instead of yoga balls. So if you think of a wall, like a big, strong wall, we want our calcium to be interlinked like a bunch of bricks within that boron rebar going through, giving it that very strong cellular wall integrity. But what can happen is if we don't have enough, maybe we have abundant nitrogen and then we're pulling a bunch of water up, is instead of this, our cell walls can be flooded and look more like this. Now, which one of these do you think a pathogen has an easier time infiltrating? Imagine me running as fast as I can into a brick wall, or me running as fast as I can into a wall of yoga balls. It's like the wall of yoga balls is going to be infiltrated significantly easier. So that's why having calcium at those critical times, especially in fruit development, especially just throughout the growing season, making sure that our calcium assimilation is keeping up with our plant growth, is very important for our cellular wall activity. Now we're going to talk about how the plant takes up calcium. So I'm going to draw a really bad drawing of a plant. So here we have our plant. Now, we all know that plants photosynthesize and they breathe. So they take in CO2 and they get rid of oxygen and water through transpiration. So we have water that goes up the plant and it's sucked up through this difference in pressure, where when the stomata of the plant are open, it's letting things out and that's pulling water up. Calcium's uptaken through that mechanism, so it's uptaken passively, meaning that it can only be taken up when that plant has its stomata open and we have water flowing up the roots. So we have this calcium 2 plus ion. Our plant is breathing, the stomata are open, and it's photosynthesizing, and it's sucking water up. This calcium ion is dissolved in that water, and it's going up the roots and into the plant. Now, when it's in the plant, calcium's really interesting. It is the only macronutrient that's immobile. So calcium only moves up through the xylem. It can't be redistributed like we see with nitrogen. So with nitrogen, there's a deficiency. We see it at the older leaves first because the plant is pulling from that reserve. Now, when calcium is needed at new growth, it can't pull from the other leaves. It can't enter into the phloem and then be redistributed. It has to be taken up continuously through the xylem, through this passive uptake. Now, that means that when we see calcium deficiency, it's going to be in this new shoot. And so what can happen is, say you have a really cloudy day or a bunch of rain, and your plant is growing and it's putting out this new growth, but it's not transpiring. It's not pulling water up because it's so humid and the stomata are all closed, and so it doesn't have this difference in pressure, and so that passive uptake from calcium can't go up into the plant. That's super important when we're thinking about fruit production, because what that means is if we have a period of significant fruit production, it all has to be passively uptaken to build those cell walls. It can't be redistributed in order to build the cell walls for that fruit. So if we have a period where we have rapid uptake in fruit enlargement, and we have low transpiration, low calcium uptake, maybe there's something going on the soil, which we'll touch on later, that's preventing that uptake, then we're going to have a lot of issues with cellular wall integrity. We may even see some deficiency signs at this young leaf. And so that's super important to know how calcium is taken up and how it's not moved around in the plant, it's immobile. That piece is really important when we're looking at something like a sap test. So when we're comparing a young leaf and an old leaf, we want to know that that old leaf has the amount of calcium and it's not going to be moved around. Whereas if we look at a sap test with a young leaf and an old leaf with, say, nitrogen, and we know we're about to come into a period of high growth at a critical point in the season, and in nitrogen, if our old leaf has a lot, then we know that that's our reserves for this young leaf. That's not the case with calcium because calcium is immobile. So when we're looking at our sap test, we're looking at this young leaf to make sure that it has enough, and maybe we need to figure out a way to get it more if it's too low and not rely on being supplied from older leaves because calcium is immobile. So to reiterate, calcium is taken up through passive uptake when the plant is transpiring and sucking water up with that difference in pressure between the root zone and above the soil. Then when it's in the plant, calcium is immobile, meaning it has to continuously be taking nutrients up from the roots or through a foliar spray. Through the foliar spray, this is a quick note, is if it's immobile, it may not make sense to do something like a foliar spray. I'm not 100% sure on this, but if we're thinking about it, that it's immobile, if we do a foliar spray on the leaves and it gets assimilated and it can't move, how would it get to the fruits if we're in that period of enlargement? It seems to me that it would make sense that a fertigation or something through root uptake for the specific reason of building fruit cell wall integrity may make more sense just because it's immobile. Cool, so that's how calcium is uptaken. Now we're going to go over calcium soil dynamics. So calcium is a part of your base saturation percentages, which as a quick overview are all of these cations. So all of these sit on your cation exchange sites in your soil. So clay has these electrostatic charges, clay is negative, these ions are positive, they hold on to each other. Now, we want to make sure that these are all in a correct balance because a key important piece of calcium uptake is that it must not be inhibited by an abundance of another ion at the root zone. So if you have a ton of magnesium right at the root zone, it can outcompete due to its charges with calcium and prevent that root uptake. Y'all have probably heard about flocculation versus dispersion. And so that creates soil structure. But why is that? Why does calcium help flocculate soil? We're going to talk about this idea called the hydrated radius. So we're going to get a little technical in the chemistry here. So we have a calcium ion and a magnesium ion. Now, when they are hydrated, so when they have water, they have different radii. So they're different size particles. And that determines how they interact with the clay particles in your soil. The calcium hydrated radius is small. The magnesium hydrated radius is significantly larger. That means that if we have clay particles that are trying to electrostatically connect with these ions, for magnesium, they're spread apart. So this 2+ and this clay minus has a further distance, and so it's not as strong. Imagine taking two magnets, positive and a negative, and you can put them together. Now, imagine there's a big pillow in the middle between those two magnets and try putting them together. Those magnets might fall. That's why an abundance of magnesium can cause dispersion, where those clay particles aren't aggregating, they're not coming together as well because this hydrated radius is larger. It's that pillow in between those two magnets. Whereas with calcium, the clay particle is way closer, and this causes flocculation and creates the soil aggregates to prevent soil from being easily compacted. So when you have a lot of soil aggregates, it clumps together and you have more air pockets, so you have better oxygen in your root zone. And so you have decreased levels of anaerobic conditions and all of the negative things that come with compaction. So that's why an abundance of calcium or having enough calcium causes that aggregation, and why having too much magnesium in your base saturation can cause situations where your soil is more easily compacted. Now we're going to talk about a couple other soil dynamics that can prevent calcium from being uptaken by the plant. So we know calcium is uptaken in this form. This is the ionic form. However, when we have different pH levels, or we have different ratios of certain other ions in the soil, they can bind to each other and cause what's called a precipitate or an insoluble form of uptake. The most common that we know is lime. So we apply lime to raise our pH. So why does that happen? We have lime, calcium carbonate, and in an acidic soil, we have an abundance of these H plus ions. And what happens with having this acidity, right, pH, potential of hydrogen, here's our hydrogen, is we get a calcium ion and a bicarbonate. So that frees up this calcium to be able to be uptaken into the plant, and it also switches this hydrogen from making your soil more acidic to more alkaline with this bicarbonate. So that's how that works. Now, this is not water soluble. It must have this free hydrogen ion in order for this reaction to occur and to raise the pH of your soil and make this calcium available, like plant available. Now, that's not the case with gypsum, right? So gypsum, calcium sulfate, is water soluble, and that can separate, and then you have sulfate and calcium, and that calcium can be uptaken by the plant. Now, if you have soil that is more alkaline, above 7 pH, you can have phosphorus precipitates. So say you apply an abundance of phosphorus right at the root zone, what can happen is your free calcium, especially in calcareous soils that have high pH, can interact with that phosphorus, right, this phosphate ion, and it can make these insoluble compounds like this. So this is not plant available, and it makes it way harder for your plant to uptake either calcium or phosphate. And so we want to avoid that. And so that happens when your pH is too high, and it causes these precipitates, and it makes it harder for your plant to take up the nutrients that it needs. So with all of that, we can now look at base saturation. It's known that the ideal levels to prevent all of the things we talked about that can prevent uptake, the ideal levels are roughly 60 to 70% calcium, 10 to 20% magnesium, and 2 to 5% potassium. And so it may or may not make sense for your specific operation to adjust these. If this is, I've seen soil tests where this is like 40. Obviously you want to try and get that down, but it may not have an economic response. But that could be what's causing potential compaction through that idea of dispersion. So getting your levels to be fairly balanced will help with that soil aggregation and your overall soil health, have a better home for microbes, and have an easier time for your plants to be able to uptake the ions when they need them. So that's important for what we talked about earlier with that cell wall integrity and making sure your plant has the ability to talk to itself with that really cool internal secondary messenger idea that calcium does for the plant. So with this base saturation, these are the numbers that we want to hit so we can increase calcium uptake to the best of our ability and have the least compacted soil, best home for soil microbes, and just have that balance of air and water in soil. All right, now to recap what we talked about, the two functions that calcium does is the cell wall integrity and being the internal secondary messenger in the cells of your plant so it can talk to itself. Calcium is taken up passively, driven by the pressure gradient that happens when the plant transpires, or when the plant has its stomata open and it's breathing and causing that pressure to uptake the water from the root zone with those calcium ions in it. And so calcium is taken up in this form, and then when calcium is taken up into the plant, it gets locked into one of these and is now immobile in the plant. When calcium is in the soil, it can react with phosphorus to make things like calcium phosphate, which is not able to be uptaken by the plant. And it has ideal ratios in the soil of 60 to 70% of base saturation. So if this number is too high, like 85 or something like that, your pH can go too high, and then you start having problems with calcium precipitates and it not being able to uptake other important elements like magnesium and potassium. And so this is overall calcium, what it does in the plant, how it's taken up, and how it interacts in the soil. I hope you learned something from this video. I had a great time putting this together. My name is Jack again. I'm the founder of Cultra, a nutrient analytics platform where we've taken all of your tests. So we take in your soil tests and we compare them to your tissue or plant sap tests so we can figure out exactly where your problems are, if you're seeing calcium deficiencies or something like that for every single nutrient. Thank you so much for watching.